My great pleasure. Well, half of you have already met Joseph on the tour, but to introduce Joseph Brunet, who's the research fellow at UCL and leading up the activity for UCL at ESRF on HIP-CT. He's now going to do a highly truncated, what was supposed to be an hour demonstration, half an hour, because you have so many great questions on the tour. Overall demonstration of how the data can be used, downloading. Please, Joseph.
So yeah, hello everyone. So yeah, I will go quite quickly to show you talk a bit more about the data. So here is kind of a rough pipeline. So you saw the sample preparation in the lab, you saw the beam time, the scanning of the organ at BM18, and the two steps that you didn't see is reconstruction and data pipeline. Just 2 minutes on reconstruction. I will go very quickly and don't go into detail. So if you have any question, you can come to any of us to ask after. But you can see here the whole pipeline that we have. So we have two ways of scanning, one for very large organ, one for a normal way. And after that, we have a step of background subtraction. Then we have the classical Paganini filter back projection where you reconstruct your slices. And finally, we have post-processing where we stitch all the scan to have the complete volume of the organ. And then you have ring correction and data reduction.
Laser pointer and forwards, backwards. Laser is the top, and forwards on the right.
Let's go. That's nice. Thank you very much. So reconstruction, and after once the reconstruction is set, you have your volume, and you can start to work on your data. Just a quick introduction. So when we speak about the volume, so here you have a complete heart. It's a nice image, but actually, how do we work physically on this? How does it present itself? It's a set of slices. So you can imagine your heart is cut into many slices like this, and you will have a folder with slices number 0, 1, 2, 3, and we have, for this one, I think it's like 10,000 slices. You can see that they're all in JPEG 2000. So we tend to reduce the data. Usually we get them in TIFF, but we convert them in JPEG 2000 that allow us to gain a factor 10 in size, which is quite efficient because, for example, this heart is 1 terabyte of this size. And with JPEG 2000, it's 100 gigabytes. So it's easier to share, it's easier to transfer, and it's take less impact on the system. Still on the data size. So here is a graph that we did on the volumes of a whole organ. And you can see the resolution here. And here you have the data size in gigabytes, and it's in logarithmic scale. So you can see that when you increase the resolution, so you decrease the pixel size, your data size increases very rapidly. And you have different organs. They are all following the same curve because they are limited by the jars, actually. So in routine, what we do is we image the organs at 20 microns now. So it's around 1 terabyte of data for a brain or a heart. We already image at 8 microns the heart and the brain, but it starts to be 15 terabytes of data. So it's quite difficult to manage. So yeah, so that's something to think about if your system is capable of handling the data, how to work on this. Yeah. So once you have your volumes reconstructed, they sit on ESRF repository, and we have 3 ways of sharing them. The first one is to collaborators, where we use Dropbox and Globus. So we share the raw images, and they can do analysis on them, classifications. We have another way, which is NeoGlancer. So I will show you that after. It's a web-based interface where you can explore the data with a normal computer. You don't need to have a very high-end computer to look at your data. And finally, we add them to the public website, the HOAHub website. But we do that after we've done all the analysis, all the quantification we want, and we publish on the data, and after it has to be added on this website. So I will present you this first NeoGlancer. So here you have an example of NeoGlancer, but I think I will show you in live. what it looks like. If I have it here, yes. So here is a lung. The data size is around, I think, 300 gigabytes. But here I'm looking at the data on my 16 gigabytes RAM computer. So normally I shouldn't be able to open this data, but with this tool, I'm able to zoom in the lung and open the data. So it's in a N5 format. So it's a hierarchical format where the resolution you see is adapted from your zooming. So the more you zoom, the more details you will see. So if I zoom very high, normally the resolution will adapt. Yeah, exactly like this. And you can see here that you have all the different bins. So here full resolution, and here is quite binned. And what's cool with this is web-based, so you can quickly share it between collaborators, and you can quickly do analysis and look at interesting areas. You can even do annotations, where if I succeed to make it work, yeah, for example, here, you can do like, maybe I will dezoom a bit first. So if I, let's say this, I want to look at, no, we'll say this artery, no, this airway. We'll do this like this. So you want to select this part. You will do the annotation like this. You can do a description, like I write a bunch of nonsense. And what is cool is once you have done that, you can copy the link, send it to your collaborator. So we'll just say like you open a new window. And normally if it works, yes, it's exactly in the same place with the same annotation that you write. So you can quickly, work between collaborators, do annotations, write label stuff, and you can quickly work like this. So it's very nice. And you can work like this on very huge data sets, so it avoids all the transferring of the data between collaborators. So this is one of the tools that we use quite a lot. Up. The second thing that is quite important is the website. So you have Human Organ Atlas website. You can Google it, and on it you have different patients with different diseases that you can explore. On each of these patients, you have the organ that we scanned, and if you select an organ, you will have all the data set related to this organ. So if we go, not on this one. Yes. So if you do Human Organ Atlas, you select the first one. You would have the website with all the a lot of information related to the project, the funding, collaborators. So you can read this. You have a help tab to help you with like dealing with the data and some information, how to download everything. And you have a search tab where you can, so you have explore first, like you saw on the slide. So you can select a patient, then you can look at different organs that we scan. So you have left lung, right lung. This one is quite straightforward. You select a lung, and then you have all the data set that we image on this lung. So you have the complete, this one is the complete organ. So you will have the complete lung lobe. And then you have zooms inside, and you have the resolution here at which we scan this organ. So you have 6 microns and mainly 2 microns with a small description of what was scanned. So here you can see that there's a lot of data sets. So this one was a COVID donor lung. But you can also go to search. And in search, you have some filters where you can filter so the sex, you can filter the age, you can filter the heights and the weights, and per organ also. And so let's say we want to select this data set.
Can people try this in real time nowadays?
Do you think so?
No, I'm just asking.
I see no one with, yeah. You could try in real time also.
I'm not going to be as fast as you are.
Oh, sorry, I have to go faster. And so you have all the data sets, so you have all the information related to these data sets. You have a description, you have the DOI, the user technique instrument, so the beamline. You have also a NeuroGlancer. So I think there's only on this data set for now the NeuroGlancer, but we are in the process of adding the NeuroGlancer to all the data set on the website. And after you have all the metadata related to the patient, the sample itself, and then the scan parameter that were used to image this volume, and the processing also. And so you have a video of the slices if you want to quickly assess. So my computer is and with the Zoom, the connection is not great, but it works quite well. And you have all the volumes here that you can download. So there's the volume in full resolution on the top here. And then you have bin 2, bin 4, and bin 8. So every time you bin, it means you downsample your volume, and you gain a factor 8 between the bins in terms of size on your disk. So if, for example, I want to work on this data set, I will download like the smallest one to go faster. So it downloads. Normally it pops up a small window at first where it tells you which publication to cite to remind you, and also that you have to adhere to the rules that it's CC by 4 license. So once it's downloaded, okay, it's somewhere. You can trust me, I downloaded it this morning, and now it's there. It just appeared here, and it's already decompressed. It's amazing. And so the first thing that you would like to see is you would like to look at your data. So the first tool that I want to show you is ImageJ, or VG actually. So it's an evolution of ImageJ. It's quite used. We use it all the time to assess the data quite quickly, and it's quite powerful even if the interface seems rough. You can download it on internet quite easily. You type VG download and you will find the first website will be this. And it also allows you to look at the data. So we put the data in JPEG 2000 on the website, so it's not a common format. So you will need to use a plugin to read it. But how it's done is you would go to update in Fiji.
Crash.
It will crash normally, like in all presentations. I checked just before, normally it doesn't crash. Yeah.
So your glass uses a ZAR 9 file format.
It's N5 for us, but yeah, we're trying to move to ZAR. It will be the goal. Yeah.
Because then in data viewer could in principle be ZAR.
It could also, yeah. We didn't try because the data size will be massive.
The problem is that ZAR is single hierarchy in current implementation in Euroclassic, so we need a multi-hierarchy. As soon as that's done, we'll flip to ZAR.
Because then you could jump the fact that the user would not have to download anything directly.
Yes, absolutely. It's a goal.
Yeah.
Very good suggestion. And please ask for multi-levels. Here's what people ask.
Yeah, that would be great. This type of format is quite evolving. There's a lot of discussion at the moment, and yeah, us too, we are trying to like assess the best and to see which direction we are taking.
So if anyone has suggestions of even better tools, please speak up. We're very happy to change.
And after that, you go to manage update websites, and you can download some packages. So for the well, I already downloaded it actually. For the opening of JPEG 2000, you will select bioformats, which is a package to open different type of bio images. And there's also, I will show you quickly a small script to do a small analysis. So you would need BoneJ also. Once you have selected both, you do apply and close, and you do apply changes. It will update and ask you to restart ImageJ. You restart it and you have your plugins. So now that you have this, you can do imports. You go in bioformats that you just downloaded. You go in your folder and you select the first image. I will select this one. It opens a window with a lot of parameters, but you just have to select group file with similar names. And there's also a tool that we use ImageJ quite a lot because it has a nice tool, which is use virtual stack. So normally, if you want to open a volume, you will open the complete volume, and the volume will sit on your RAM of your computer. But if you have a computer that has not a lot of RAM and you need to open a big data set, you can use this option. So it will just open the image that you are currently seeing in your stack. So you can go and go through your stack, but it will just be a bit slower, but you can open massive data sets. So with my computer of 16 terabytes, I can open data sets that are 1 terabyte of size. And so it allows you to access quickly without having to download everything. So you click OK. OK again. And it does nothing. Sorry?
You didn't select virtual stack at the end.
No, I didn't select because this one is very small. I took the smallest one. If I would have downloaded the full resolution, I would have selected it because it's too much for my computer. And so it's downloaded. And so here you have your stack. You can use the arrows, so top arrows to zoom and right and left arrows to go through the images. To change the intensity, the contrast, you can go in image, adjust brightness contrast. You obtain this window, you do auto, and you obtain a better contrast. And then you can zoom in. So this is very bad resolution, as you can see. It's the same organ that you saw in your Glancer. But here I download it in bin 8, so it's not a great resolution, but for the sake of the time, we go like this. What you can do, okay, remember I wrote things that we could check. So we could check the histogram of this. It depends what type of analysis you want to do, but there's a lot of tools on ImageJ that you can check. You can look at the histogram, for example. So we just want to do it on this image, not all of them. So you will have the histogram of your sample. You can also do you can look in orthogonal views if you want to have overviews. No, I have to click on this one first. So stack orthogonal views. So it shows you like the different views, and you can move like this towards your stack. Yeah, there's some issue with displays, but you can move and go through your data set to assess the different views.
I think a trick for everyone is use a massively heavy bin. Just try it first. And
Yeah.
Figure out what you really need and then open up a higher risk.
Exactly. ImageJ is for that, it's a quick assessment of your data set, and you can do quick things. And after, if you want to go in deeper analysis, you can use like a higher software, or like you can use Python if you want to automatize everything. But yeah, it's a very powerful tool. You can also, if you want to quantify, for example, diameters of airway, you can draw a line and you can go in analysis measure. So it will give you the length in pixel, and you just have to multiply by the pixel size to have your length in real units. So here, how does it do? I don't know, 0.2. So it's like 4 millimeters, something like this. And so, yes, you have this, so you can do that. And you can also, yeah, we'll present this small analysis of BoneJ. Just to show you an example that is where is the script? It's there. Yes. So a quick analysis that you can do. Here is a simple script that uses BoneJ. So it's a plugin of ImageJ that was coded. You can see here. We can send it to you if you want, actually. and you run it, it will ask you an input directory. I will select raw data control. So raw data control is stack. So here on the same data set that we saw before, here we cropped a small area, and you can see the on a zoom actually of the data set, and you can see the alveoli that are here in the lung. And if you want to quantify their, like the number, the connectivity, the thicknesses, and stuff like this, we can use this plugin. So you run, you put the input directory, the output directory, the file suffix, click OK. Now it does many things. You have to put the results. There's a bug, that's nothing. We don't look at this. It doesn't exist. It works anyway, but you can see that it's segmented. It segmented the alveoli here. So you have a binned image, a bit binary image with the alveoli in white and in black the boundaries. And it calculates a lot of different parameters like the connectivity and other stuff that I don't know.
Or the characteristics.
The characteristics.
And the thickness.
Exactly. And now it's calculating the thicknesses. So it's just a bit longer. So yeah, my computer is not very powerful. So it takes a bit of time. But yeah, it's kind of a quick analysis that you could do if you would like to assess. So here, I just show you the control area, but we have also a diseased area. So you can assess in a lung, for example, you saw that in some places you have a diseased area, some places you have some area that look quite healthy. And if you want to assess the discrepancy between the different areas, you can use this tool to check. Almost there. Yes. So here, I think it's a color map of the thicknesses. Is that it?
Pore size.
So there's the pores and there's the
Struts.
struts, you call it. Yeah, sorry. I call it the boundaries, the struts. And so here you can see the different thicknesses. So this is a very quick analysis that you could do on your data set to assess. If you want to do segmentations also, I have a quick example that I will show you here. I have to, oh my God, I have to reconnect it. So this image J is to quickly assess the data set. You can do quick things to look a bit, do quick analysis, measurements, stuff like this. And after, what we do is when we want to quantify better some morphological structure, we use other software. So here it's VG Studio Max, but you can also use Amira. You can also use Dragonfly. It depends on what your lab is using. But they all have kind of the same tools. They're implemented a bit of a different way, but they all work the same way. So here it's a heart. And you can see that you have it in 3 different orientations. So here I will show you how to segment a bit some arteries. So for those who knows a bit about this, you have the aorta here, and here you have, I think if I go through, yeah, you have the coronary that starts from the aorta and that goes supplying the heart. And so if you want to segment this, you have many different tools on the top. Here you can draw, but it will take you a long time. What we use a lot here is a region growing algorithm. So you click on a point, that's it, and after the segmentation is transmitted toward the other pixels around. And if between 2 pixels, the intensities are over a threshold that we call tolerance here, it doesn't propagate, so it stops. So it stops at the boundary that you set. So if I use, for example, 500 here of intensities, and I start here, you can see that it goes in the artery. So if I do it a bit smaller, like this, and you can see in the different views. And if I put a bit more, because here it doesn't go out, it's a bit better. You have less holes. But if you put too much, you can see that it goes out, and so it's not good. So we would like to stay at 1000, for example. You create your region of interest, and after it's bounded, so it's bounded by a sphere, so it doesn't goes everywhere. And after you follow your artery, and you can continue like this. You can add it to the previous region, and you can look here, you see nothing because I didn't enable this. So if I don't make it transparent, you can see it here appearing. And so if I continue a bit further, here, for example, just to show you. And I add it, look. Wow. And you can continue like this iteratively and segment the whole thing. And if you do it during a certain amount of time, you can obtain something like this. So here, if I put it a bit bigger, yeah, you can see here the aorta, and you can see the arteries in red and the veins in blue. And you can see it's the heart, so you have the shape of the heart. So this will take, with this software, it will take approximately 2 or 3 days, something like this. But it depends, the time will depend like how precise and nice you want your segmentation. If you just want a visualization, or if you want something that you can quantify, do skeletonization and stuff like this. So yeah, here's a good example. And I think with this, I have presented everything. Yes, and I think I'm right on time.